it's my first QS. I just moved to Berkeley 3 months ago, and I just found out about Quantified Self when I got here. So I thought, okay, I really want to go. I don't really have any data. What's the biggest repository I have? Google Calendar. I don't know how to download data from there. I can't plot it, so I was like, okay, I need to think of something else. So I work in I came from London. I work in a brain stimulation lab, and now I'm in Berkeley, and I work in a brain and sleep imaging lab. So I thought, okay, my brain's probably the best data storage device, so I'll just show you my brain today. So here's what I mined from it. Okay, sorry. This is up here. So I look at a lot of brains all the time, and they start to look the same. Like, it's really boring. And that one's mine because I only know because I colored it in. And so, but I'm pretty sure my brain's kind of different from other people's. You know, I have to be special, and you know, every brain is different, slightly different, but how can we find that information? And there's a lot of information stored in the structural anatomy of our brain. We can know things about our personality trait, you know, things related to like neuroticism, anxiety, extroversion. Someone was talking about Myers-Briggs just now. And there are other types of higher level cognition as well. So we know this simply not even from activation, but from our neuroanatomy. But these are all group studies. So this doesn't tell me anything about myself. I can't extrapolate anything meaningful if I want to find out something about myself. And so, you know, I wanted to take a look inside my brain, and I wanted to see what makes it special. How is it different from other people? But these quantifiable attributes, they're hidden in the raw data. So you have to run statistics in order to try and tease them out. So what's inside an MRI brain scan? Let's like debunk this right now. Functional MRI measures bold signal. That's blood oxygenation level. So it measures how much oxygen you're using in your brain. But what I want to look at is structure. I just want to look at neuroanatomy. And so structural scans, or T1 weighted scans, they are sensitive to different tissue densities, your gray matter tissue and your white matter tissue. And we're using a statistical protocol called voxel-based morphometry. And it looks at density of each voxel across a bunch of brains, a bunch of people. And it looks at inter-individual variability in tissue density. So what I looked at was how my gray matter concentration was different from other people's. So how many standard deviations does my gray matter concentration, you know, how far does it lie away from the population mean? And so we need to do some standardization. I mean, I'm sure a lot of you know what standardization and z-scores are, but I'm just going to go through it really quickly. So to calculate a z-score, you need to take the mean, you need to subtract the mean of all values from your raw voxel value, and you divide it by the standard deviation. So for example, imagine I have 100 people, and I normalize it so that the mean is at 0, and your z-score is basically a measure of your standard deviation, how far you are from the rest of the population. So you can imagine the area under the curve. If you're within the bounds, if you're within the bounds of 1 standard deviation, then you're within 68% of the population. And then if you're further out there, then that's roughly 95% of the population. But you can also look at it this way. If you're 2 standard deviations above the mean, that means that you're 97th percentile. So you're above 97% of the population. And yeah, so that's roughly how it goes. So that's exactly what I did. I looked at my brain relative to 106 people. So it's roughly men and women, half-half distribution. And they're mostly 20-something-year-olds, so I'm 25. Kind of fits my population. These are all from London, because I haven't collected any brains here yet. And so here's roughly the algorithm or the protocol that we do with the raw image. First, you need to segment the data. So a raw brain image contains a lot of information, right? It contains white matter density, gray matter density, and cerebrospinal fluid. So you need to separate it. And what we're interested in is gray matter, because that's where your brain cells are. And so we're only looking at that. And then after that, you need to say I have 100 brains, and I need to all put them into like a normalized space. So I need to fit them all into the same space so that I can compare them, because each of our brains are different shape and different sizes, right? So you need to estimate the optimal deformation by iteratively fitting them to the average. And then after that, you generate the actual spatially normalized images, and then you can run statistics. So I can calculate my z-scores of my gray matter concentration after doing all these image pre-processing. So here's what I found, and this is kind of scary. It's really, it's really revealing, because I only did this weekend, and I'm showing you things that I've just learned about myself, too. And I really believe that your brain structure is not going to lie to you, so we'll see, maybe. So based on my brain's volumetric distribution relative to other brains, I'm going to show you the good things first. So here's my brain areas that are 1 to 2 standard deviations above these 106 people. So that means this is like 84 to 97% above the population mean. So that's like, that's where I lie. So these are brain areas. So my motor areas are pretty good. My superior frontal gyrus is the opposite of schizophrenia. So schizophrenics have decreased gray matter volume in that area. And people who drink a lot also, they show a little bit of superior frontal gyrus decreased volume. So I don't drink much, and I don't know, maybe. My executive attention area is pretty good. So that's, like switching tasks and, calculations and things. And my selective attention is very good. So apparently I'm very attentive. And then my angular gyrus. So some people call this the Einstein place. I'm not sure. I don't really want to blow my own horn. But it's for metaphorical and abstract thinking. And then my right hand area is pretty good. So I'm right-handed. I don't know why it's higher than other people, but I'm not like super dexterous or anything. My auditory cortex is really good. I listen to a lot of minimal. I don't know why. Maybe that has to do, or I'm a good listener, maybe. And then that's my mid-temporal gyrus. It connects to the face area, but again, in schizophrenics, this area has reduced volume. So mine is has more gray matter volume than other people. I'm not really sure what that means. And then my inferior temporal gyrus, it's really good for semantic memory, visual perception. It's called the associative cortex. So apparently, that's good for me too. And again, I'm not schizophrenic, so this is all pointing to the same direction.
What's the 10 to 20 scale?
The 10 to no, it's 1.0 to 2.0. So that's
okay.
1 to 2 standard deviations, and that's what the color scale is. So again, more angular gyrus, abstract metaphorical thinking. And my intraparietal lobule, that's good for number processing. I think around this time I was starting to do a little bit more programming, so maybe that's why. And then so this area was what I liked. I was like, oh, I'm so empathetic. I have really good interpersonal competence. Wow, amazing. But it's actually really small. So that's on the right side. On the left side of that brain, that's your language processing, but mine wasn't very good. Even though I'm bilingual, I thought I would find something, but I guess a lot of people are bilingual in London because we don't have any Londoners anymore. Yep. So let's take a look inside. That's my caudate nucleus. That's subcortical. And that's really that's useful for sensory motor coordination. It's part of your motor area again. So it makes sense because my motor areas are really, they're great. So they connect to these a lot, and you'd get the same pattern in the same network. Correct response selection. So this is the area that tries to select the correct response that you're meant to make based on your goals and based on, what would happen. Maybe I'm good at that. I don't know. And then here, my hippocampus. Not really. I wished it was, more yellow, because so hippocampus for memory. But I don't read a lot. I don't learn that much all the time. So, and then this was interesting. So the mid-cingulate is responsible for a lot of things, but this area specifically, it's kind of more sensitive to pain and heat, and I'm so sensitive to temperature, and I think maybe that's why. And this part, precuneus, self, consciousness, mental imagery, I started meditating when I was 16 years old, so I think maybe that contributed to it. And then the thalamus, this is your sensory gating center. So maybe I have, very good sensory gating. I'm constantly bombarded by sensory information. Like, I really don't understand why, but this is what it's telling me. So I think the interpretation is a little bit hard. So those are all the bits that I have more gray matter concentration than other people. Now let's see what I have less. Okay, so this is the back of my head, my whole visual cortex almost, and my cerebellum. So I've had these glasses for about 7 years, and I wear them less than once a week. And seeing this has made me really want to wear them a lot, because I'm meant to wear them every day. So, this is crazy. Cerebellum is for movement, maybe some timekeeping. I'm late all the time. I don't know if that matters. And okay, so here's something really interesting. Tool use. This is an area associated with tool use. I tried to fix my bike yesterday, and I've stopped riding it because I can't lock it. I don't know how to use the lock. So maybe I need to improve on my tool use. And in men, this area is bigger. And then this area was really interesting. So that's responsible for social concepts. And in schizophrenias, they have reduced gray matter concentration in this area, and it's associated with their hallucinations. So sometimes, this is really revealing, you have to understand. I do sometimes have weird imagery. I do sometimes hallucinate. And sometimes I'm socially not quite correct. And so more emotional regulation and inhibition. That's also my frontal area. During this time, I was working really, really hard, and I wasn't really being social. I was like living under my desk because other Japanese colleagues were doing that, and I thought that was what we were meant to do. And I was like really anxious. I didn't know what the heck was going on. I was like a teenager basically, and I would like burst out at people. So this was really interesting to look back, and I get more emotional regulation bits. So yeah, this was a real learning experience for me. My footwork coordination's not good, apparently. That's my foot motor area. So, no square dancing, I don't know. And my superior colliculus, that's for controlling eye fixation. So that's the last one I'm gonna show you. And actually, I want to quit this for a second, and I want to show you someone else's brain. So this is my colleague's brain. It's better in 3D. But it's really interesting if you know the person. So his pattern, red is higher than normal, and then green is lower than normal. His pattern is totally different from mine. So he has a ton of stuff in the temporal lobes. He learns a lot. He reads a lot. He has a really, really good memory. And it's really interesting when you know the person and you look at their brain and you're like, hey, yeah, this kind of describes who they are. Okay. So, sorry. Let me go back. Okay, so the scan was taken last winter, around maybe January, I think. And so, a lot of time has passed. My lifestyle has changed drastically. I've been meditating more and sleeping more. And so, maybe my emotional regulation is better. It is better behaviorally. And I'm curious to see what it looks like in my brain. Maybe I need to start wearing my glasses more. And I may start to take tennis classes to take advantage of my, apparent motor skills. So we'll see. And so my vision is, basically, I wish that everyone in this room can look at their brain. Because the feeling of seeing that, it's like, oh wow, oh my gosh, is that real, And there's so much information in there. You can correlate it with anything you want. So a bunch of colleagues and I, we're basically building this. We're gonna build this. We're gonna try and launch it maybe in spring next year. And it's gonna be basically a place where you can upload your brain. where you get your brain is really simple. You go to a research facility, go to UCSF, go to Berkeley, get paid to get your brain scanned, and then tell them, give me my brain. You know, like, it's my brain. Give me my brain scan. And then you can upload it. And then all the stats is done for you. And so you can imagine, if there are 10,000 people on this database, you can say, I want to compare my brain to neurotic teenage girls. I want to see how far I've actually come from that place. Have I really moved on? And you know, you can correlate it with your scores on lumosity and test my brain, quantified mind. And if you have, your genetic blueprint, then you can look at other people and see how it compares to yours. You know, I want to know how my brain compares to Lady Gaga's, for example. Like, she's amazing, even though we haven't heard from her for a while. And so the goal is repository building. The more people do this, the more useful and the more information we can get. And so the more meaningful things that we can get out of our brain data. And so this is the public library in Stuttgart. It's amazing. And I often see our brain as that. It's just a big library of a lot of books. And when you sleep, you know, that's like when creativity, like, things get primed. You have like a lot of associative network reorganization. And so same as neuroplasticity, it's not a myth. It's like, it just takes practice, it takes behavioral change. And rearranging is faster than regenerating. We do grow new brain cells, but rearranging is faster. It can take, you know, a week. And so it's real. It's changing your behavioral patterns to create physical change in your brain, in your physiology. Yeah. So I hope people are psyched about this because, you know, it's a new way of profiling. Thank you.
Okay, so somewhere around a bit over 2 questions, I'd say.
All right, cool.
Cool.
Yeah.
I don't know.
Pick away, yeah.
Yeah.
So on the graph you're showing with your brain and the different colors and the sigma that you had, the one was that,
Yeah.
is that right? 1.0 to 2.0 is sigma.
Yeah, that's
was
the
relating to
design
some population data.
Yeah. So remember I showed you the pie chart with the 2 graphs. That's the population density. So it was like 109 people.
So the 109 people data. I mean, is that data specific, like genre or type, or I mean, is it?
I mean, I only had age and gender there, and they were, you know, the mean age was like 26 years old. So it's not a lot, because the older you get, the more morphological changes you get in your brain structure.
Right, because I mean, you were making basically statements about the sigma based on that population. Right? It's not related to yourself. It's not, you know what I mean, like as a reference point.
Yeah, it's my Z-score relative to this population.
To that particular population.
Yeah.
Okay.
Yeah. So that's my Z.
Yeah.
Yep.
You talked a lot about how this brain density of different areas of the brain changes through behavior or different circumstances in your life.
Yeah.
But how much of it is genetic or not changeable?
Well, so the gross structure of your brain is very fixed. Like, your occipital cortex will always be your visual cortex unless you become blind at a very, very young age and then it gets rewired. So we're talking about very small changes in capacity, but the small changes, they come through in your behavior as well. And it's actually really reversible. So if you can practice juggling, for example, practice juggling for a week, and you know, your hand coordination area will show very, very slight changes. And then say you stop for 2 weeks, it'll go away again. So it's super plastic, it's fast. Yeah.
Sometimes it's more the connections, which are more the white matter than the gray matter, and I noticed you concentrate on the gray matter.
Yeah, so you can run the same statistical test on white matter density as well. So I haven't done it because I just ran it on gray matter, but you can do it on white matter as well. And in fact, you can look at something called fractional anisotropy. So it looks at like water diffusion in your white matter tracks, and that's a better measure of the wires between your brain cells, if you like. Yeah.
Okay, that's a lot of questions. Sorry,
okay,
guys.
yeah.
One more round of applause.